Hello everyone, I'm Katia Ahmed, a PhD student at University College London, and it's my pleasure to welcome you all to webinar number 13 of the Comp Biomed webinar series, run in collaboration with the VPH Institute. Comp Biomed is an international centre of excellence dedicated to the advancement of in silico medicine, working towards the full realisation of the virtual physiological human. So today's talk is about some of the ongoing work to tackle COVID-19 that's currently underway at Argonne National Laboratory, with a focus on their efforts to apply machine learning techniques to discover more about the interactions between small molecules and the novel coronavirus proteome, and to accelerate multiscale simulations of these proteins. The talk will be delivered by Dr Arvind Ramanathan, who currently works as a computational biologist at Argonne National Lab and is a senior scientist at the University of Chicago Consortium for Advanced Science and Engineering. His research interests lie at the intersection of data science, high-performance computing, and biomedical sciences, with a specific focus on scalable statistical inference techniques. Arvind received his Master's in Computer Science from Stony Brook University in New York, and in 2010 he received his PhD in Computational Biology from Carnegie Mellon University in Pennsylvania. Following a stint of postdoctoral training, he went on to lead the Integrative Systems Biology team within the Computer Science Division at Oak Ridge in Tennessee, before moving to his current post at Argonne. So before handing over to Arvind, I'd like to remind you that today's webinar will be recorded, and the recording will be uploaded to the Comp Biomed website and YouTube channels fairly soon, so you can always go back and reference certain parts of the talk or pass it on to friends and colleagues. Questions will be answered at the end just to keep things running smoothly. So if you have any questions, write them in the question box within the GoToWebinar control panel at the end, and I'll read them out to Arvind. So that's all from me. I hope you all enjoy the talk. Thanks very much, and over to Arvind.
Thank you so much. Thank you, Katya. Thank you all for attending the meeting today. I'm going to talk a little bit about our ongoing efforts in terms of understanding how we can apply artificial intelligence for targeting the entire proteome. And this is joint work with a whole bunch of people, nearly 300 people, including UCL, my collaborators, Pete Conley, and a whole bunch of people from there, including Josette Haider, who is another faculty. And I'll highlight as we go along who are all the contributors to the major part of the work and so on. So everybody has heard the story about where COVID arose. It came from Wuhan, quickly spread to other provinces, and now it's a worldwide pandemic, unfortunately. And we are having to deal with the fact that this virus is quite novel. It's larger and far more stable than its counterparts, and it can live on surfaces for a while. So we are currently working with Romy Amaro's group, where we are trying to build an entire model for this virus, atomistic scale model for this virus, with all the spike protein, its glycosylations, and the RNA domains, and everything that is present in the virus. And this is a fairly large system to simulate. And so definitely takes a lot of effort in terms of getting to that point. And this is something that we are hoping to achieve as part of this campaign. And this is needed because we would like to understand what are the initiation events for this virus in order to attach and start infecting human cells. But it's not only that, we also need a comprehensive strategy to kind of go ahead and say, how do we identify small molecules or other therapeutic strategies that can include antibody design and things like that to treat the infection that's already present, or even prevent future viruses from happening. So in terms of the US alone, if you just look at the number of people that have been infected with it and so on, nearly 6.3 million people have been infected as on September 10th, 2020. And you can see that the curves are going down, but still the infection rates are quite high. With this whole situation, the importance of identifying small molecules or therapeutic strategies that go to treat COVID-19 become almost like critical. And what we wanted to do within the Argonne Lab was to come up with strategies, AI and computing methods that could fast track these things at scale. And that's one of the reasons why we chose the entire proteome, targeting at least a major portion of the proteome instead of actually targeting one or two targets that's currently done within other groups. So this project is actually funded through a consortium, which is called as the National Virtual Biotechnology Lab, which is established by the US Department of Energy, where there are several areas that they're interested in actually funding. And this is to basically aid the US policymakers in terms of responding to the COVID-19 with real-time or near real-time epidemiological information and decision-making. It's also to accelerate the production of several critical medical supplies that are needed across the nation. And we are targeting the use of AI and supercomputing to design small molecules that can target various proteins of the SARS CoV proteome. And I'm not going to talk about all these different efforts. I'm just going to focus on how we are using supercomputing and artificial intelligence for the design of these therapies. And mainly from the perspective of looking at computational drug screening, which is to say, can we design antiviral drugs by repurposing existing compounds, or basically design some new compounds that we get as an idea from the process of lead refinement and things like that. The second aspect that we are also interested in is looking at the sequences and trying to see, are there evolutionary hotspots within the virus that we can design the immune system response. And we're also doing quite a bit of analysis of the virus evolution itself in order to find out what the evolutionary origins of the virus are and kind of map out its trajectory, as you may say. So all of these areas explore the use of data science, artificial intelligence, as well as high performance computing. And we're quite fortunate. We have been blessed with computing resources across the nation. We've been able to use Frontera, which is another supercomputer, Summit at OLCF, and a whole bunch of machines on which we are being standing these infrastructures up. So the real workflow that we are trying to do is kind of highlighted here at a high level. And we are looking at various targets, protein targets, from the SARS CoV-2 as well as human proteins as well, because these are host interactions that are equally important, where we are trying to model host pathogen interactions using network-based approaches. This is across 9 different national labs, as I already told you. And so therefore it includes a whole bunch of collaborations across different labs. So we get information from that. We have the advanced photon source where many of these structures were determined. And we determined candidate targets for actually looking at good structures on which we can do virtual screening. Of course, when you come to virtual screening sort of approaches, there are already very good systems that do that. So you have drug databases like Inamine, Drug Bank, Zinc, all of which contain billions of molecules. But these billions of molecules are really not in a format that a machine learning algorithm can take and work with it. So we are doing this featureizations on the fly and things like that, which can be quite expensive. So what we have done is basically taken that entire set of featureization and everything that we need to do and made it available to the public. And I'll give you some vignettes of what's going on there in a short while. Once that happens, we take it to physics-based docking codes, which are quite efficient. We use OpenEye as a preferred tool within our group, but we have also tried Autodock as well as Dock from UCSF as our engines for doing the docking. And we'll also talk a little bit about how we are using reinforcement learning as an AI approach to model molecules as they bind to the protein and so on. So that constitutes one of our workflows. A second strategy is now to say, of the compounds that we have screened using these docking molecules, we cannot be, happy with the fact that we have just screened hundreds of thousands or even millions of molecules, but it has to go to some place where it basically allows us to say, can you infer whether these molecules would bind properly across the proteome? And that needs quite a bit of, data. for which you need to generate binding free energy estimates to kind of figure out if these interactions are stable and so on. This is where we are working with Pete Coveney's group, with Rick Stevens, and we are designing active learning strategies for virtual screening. And once that happens, we are now pushing it to accelerate the sampling to kind of see what are the substates in which these drugs are binding. Are they stable? Are they interacting with the protein in a in a manner that makes it stable for staying together? And once that happens, we actually take it to the entire scale of the virus and hopefully able to simulate as to how the virus attaches itself to the entire protein. So as you can see, this is a whole set of systems that we are targeting and working on, but this gives you the bigger picture view of how all of these things play together. And at the heart of it is a method called Deep Drive MD. I'll talk a little bit about it. It uses AI-driven adaptive sampling simulations. In one case, we are using to model scale, where we are using it for multiscale simulations. On the other side, we are using it for drug-based design strategies. So there are 9 targets from the SARS CoV-2 that we are currently looking at, which includes the main protease, the papain-like protease or PL-PRO, ORF7A, RNA-dependent polymerase, the spike protein, NSP15, ADRP or NSP3, NSP9, and the NSP1016 complex. So these are each of the individual targets on which we are working on. We are trying to look at the evolutionarily conserved residues to kind of figure out where these binding sites might be present. Are there allosteric sites in some sense? And we are going to do a whole lot of docking against them. So how does AI and ML play a role into this whole process? So the workflow that I showed previously, this is a zoomed in version of the second step of this workflow, where we take the crystal structures that are coming from the advanced photon sources, put them on these COVID-19 specific targets. We take them, do the docking across multiple supercomputing facilities, generate our training data sets on which the AI models are trained. Once that happens, we end up refining it further and further through a very simple filtration process where we take the top 2.5% or top 1% of the hits and basically simulate them using Deep Drive MD sort of approaches, as well as rigorous free energy computation methods that tells us whether these molecules are stable in the binding site. Once that happens, we are now able to give these back to our experimental collaborators who run assays on them, kind of give us a sense of whether these are working or not, and so on. So the take-home message from this is it's an entirely iterative process. We start out with some known data or some known information about these inhibitors. We look for ways to optimize them based on the binding sites that we are looking at. Once that happens, we give it to our experimental colleagues who basically do either protein-specific assays or wholesale assays from which we take the data and feed it back to our collaborators at both the crystallographic side as well as on the computing side, because we now have the ability to access what constitutes good binding versus bad binding. So let's talk a little bit about how we are preparing the data itself. So the data pipeline itself consists of developing what we call as machine readable data sets for many of these small molecule libraries. So we have about 4 billion known molecules that are all coming from these different data sets, including enamine, drug binding, drug bank, binding DB, E molecules, and all these different places. And what we have to do from each of them is to canonicalize them. So you basically get rid of all the formatting issues. You make sure that they are all represented in a similar way. Once you do that, you can compute fingerprints and do some similarity searches, which is, after all, one of the most important steps in terms of finding compounds that are active. The second aspect is to compute all these features that we care about. These are called moderate features, which we have generated. And then we can do a whole bunch of deep learning and filtering to basically say, are these molecules viable set of systems for doing that? So this work was led by Yadu and a bunch of people at Argonne, including Tom Brett and Ian Foster, where they were able to make this completely available as open data set. So the first release of these computed features for AI-based screening consists of about 23 input data sets, about 4.2 billion molecules, and 60 terabytes of data. which includes all the canonical SMILES formats, the moderate descriptors, as well as the molecular fingerprints and 2D images, which are all available on demand. So if you're interested, you can actually go and take a look at this particular website. This is very critical because AI models need this sort of data to kind of figure out what's happening. And I'll show what we are running currently in terms of some of these ideas. The second aspect, which is also important, is you have these openly available data sets, which consists of papers and literature that surveys a whole of these viruses that we know of, including SARS, the SARS CoV-2, MERS, and others. So what we were able to do was we could engage our admin staff people in the lab to extract these small molecules from these papers. And we started out with about 800 molecules or so, from which we said, okay, we can take a look at this data. And we then ran named entity recognition methods on them using natural language processing ideas. And what that allows us to do is essentially label relevant parts of these small molecules from these CORD19 papers data set automatically. And once that happens, we can actually release that. So we were able to extract over thousands of molecules from this, and you can take a look at the model as well as the molecules that we have gotten it from there in order to do some of the docking work that we are currently doing. So now that I gave you a sense of how the data as such is organized, let's talk a little bit about how we are thinking of running these simulations, right? So in order to improve the results that we are trying to do, there are two steps to this process. One is that you need to generate the 3D pose of how that molecule binds to the protein itself, which is actually given to you by ensemble docking approaches. So, OpenEye, any one of the tools that you use, they're extremely good at generating these poses. So we really do not worry too much about the generation of poses, because many of these docking algorithms are very good at doing that qualitatively and also some quantitatively. But what we allow then is, let's say I have 6 million molecules I have docked, which is fairly about a couple of hours worth of runs on a machine like Frontera. So from there, I want to be able to translate these results and see how it can go to a much larger library. So I want to say, let's say I take a library of about 128 million. So one of the first steps that you could do is predict the docking scores themselves to say, give us a sense of whether this molecule is going to really bind or not bind, right? So that idea is actually captured by this ML1 workflow, which basically tells you whether we can actually figure out whether a molecule would bind or not to the protein. And once that happens, we can say, okay, we have all these new molecules. Now generate these 3D poses from the ensemble docking again and feed it into more rigorous, what we call binding free energy estimates. So this can use Smax or it can use any one of the free energy approaches. But the idea is that we need to get something physically grounded. And this is where the binding free energy values are kind of important for us. So what happens is you are now taking the very same model that was used to predict the docking scores, but replacing it with a smaller set of seed computations that tell us what the binding free energies are. And then using a graph-based approach, we are now mapping the similarity across the compound space and then saying, which of the branches of these compound spaces can we get rid of? And I'll talk a little bit about that as we come along some of the advanced machine learning ideas there. And then once that happens, we know that Smax seed computations do have some limitations because they're not sampling all of the, conformational landscapes and so on. So in order to aid that, we actually push these simulations, the outputs from these simulations onto what we call Deep Drive MD, that allows us to characterize some of these conformational states that are being sampled. And it in turn allows us to refine the free energy computations, which can then be fed back into our prediction models. So if you really think about it, there are like two different loops that are playing a role in terms of, first identifying the binding free energy estimates and bounding them. The second one is to improve the docking itself, where we can say, okay, once we get a better docking model, we are able to do much better in terms of our overall strategy. So let me talk a little bit about what DDriveMD does. So the basic idea was to interweave simulation and analytics in such a way that we can reduce computing overheads on these machines. So the traditional way people have thought about simulations is, you have a job scheduler, you run your simulation on these big machines like SuperMOOC or, Theta or Summit and whatnot. You store the data, you analyze the data, and then visualize that. But this is unfortunately going to be a big problem when it comes to the data storage aspects when you have these new machines that are coming up, because you're going to have a lot of data movement because the amount of data generated by these machines are really large. So what if we were to remove the disk in the middle? Of course, we're not going to remove the data disk completely from the supercomputer, but what if we basically allow the simulations to be run in such a way that analysis is actually internal to the whole part of this run? And this is what we refer to as in situ analysis. And this basically allows us to reduce the amount of data movement and overheads that we have, which provides for an online mechanism for feedback and so on. So this iterative forward-backward loop kind of allows us to do this at a much larger scale than that would be possible really with a smaller set of simulations. And because we are using an ensemble-based simulation in parallel, there's a statistical approach. It allows us to do a large amount of calculations on the same system. And there are lots of questions that you want to ask about how to do the adaptivity and things like that, but we're going to allow for a very simple adaptive approach to see how it runs. So this kind of tells you how deep DriveMD really works. So we have an ensemble of simulations that are running. We collect the data and stream that data back into places where the machine learning and deep learning tasks are running. And then we run into this inference mode where we say, okay, are you identifying some states where there are novel things that are found, or are you identifying the same things that you're sampling again and again? If it's a novel state, you basically say, let's spawn off new simulations from that. If you find that the states are not novel, they're sampling the same thing that we have seen before, we terminate those simulations because you don't need to do that many times. But the important thing to realize is that there is the learning phase as well as an inference phase, which you need to pay attention to. And that can actually be on par with the same time it takes for these MD simulations to run. So there is a little bit of a, infrastructure issue that you have to innately address, because before the machine learning algorithms were much simpler. You could run them within a couple of seconds. You got the insights that you cared about from this data. But the deep learning ideas actually take as long as these simulations can run. So there's a little bit of mismatch in terms of how these algorithms need to run. So this work has been published before. If you're interested, I can also send you copies of these papers. But the idea is basically we want to minimize the effort that it needs to set up these sorts of workflows. So we have shown in the past that we can do deep learning on MD simulations. There are lots of groups that have worked on it, many groups in Europe, many groups in US. All of them take some form of an unsupervised machine learning algorithm that basically projects the data that we get from MD simulations onto a low-dimensional manifold. And this is an illustration of this manifold that you see, where each dot is actually a confirmation from my simulation, and we can paint them with some values that we get from our simulations, including fraction of native contacts or the root mean squared deviation to the folded state. And these are two metrics that you can basically say, are these looking as if we can cluster them? And indeed, you can actually see when we project them out into these low dimensional manifolds, if you walk from the left to the right, this increases the number of fraction of native contacts, which means that is actually folding the protein. And you can basically think of ways in which you can navigate this space. And so if you were to draw out some sort of a free energy profile from this, just a number to kind of say what are the minima that are found, you find that there are states that are completely folded. State 1 is completely folded. 2, 3, 4 are different intermediates that are found. State 9 is actually misfolded in some sense. And you can see that that's also present in the trajectory. But there are some intrinsic challenges in terms of saying, how do you achieve the sort of scaling that you need when you're running the simulations? So we have seen that this actually works very well in terms of folding proteins, This is the FS peptide. We ran about 840 simulations. This is an overkill. I emphasize that it's an overkill in terms of amount of sampling that we have done, but we do reach RMSDs that are very close to the native state. This is with the FS peptide example that you saw from the past. With the BBA and VHP, we can actually run much longer timescale simulation. We reach a min RMSD of about 1.8, but we can see that we can sample almost all of these different intermediates that are formed with respect to the native state across all of these simulations that we have done. So basically, if you have a large number of ensembles, you're going to get better sampling, and you're also improving the adaptivity of how this algorithms work. So it's a little bit of an issue here. I do apologize for not being able to see it correctly. But what we can claim is that with respect to traditional approaches, traditional sampling approaches, like if you have access to Anton, for example, what DeepDriveMD allows you to do is at least get an order of magnitude faster in terms of efficient sampling. So just walking you over the plot, if you have the x-axis, this is absolute simulation time versus explored population, you can basically see that if you include the learning data, there's a crossover point around microsecond timescale where the DeepDriveMD actually starts to overtake what the Anton trajectory would possibly do. This is true with the other system also with the BBA, and there is a crossover point that I just mentioned, but if you don't include the learning phase, you can kind of clearly see that this, if you have a pre-trained model from deep learning that you have access to, this change point actually happens much earlier. So this is now, I talked about the microsecond timescale, but it's about a couple of 100 nanoseconds, which is another order of magnitude speed up that you get as a consequence of doing this sort of sampling. And you can also see the quality of sampling. We are pretty good in terms of accessing all the folded states, and the coverage is quite nice. So now you get into this engineering world of saying, can we run this on the entire spike protein? So this is a 1.5 million atom system, not easy to analyze using all these different techniques. So we said, okay, we really need the help of people from NVIDIA as well as other hardware folks to do this. So this is simulations from Rami Amaro's group, which we were able to take and run it using our deep learning algorithm. This is about 1.14 billion parameters that we need to learn from the data. And you can actually see that we can scale it across multiple GPUs, including usage of memory, and the time per batch it takes is actually like going down when you divvy it upon the GPUs and so on. So this is a classic example where model parallel and data parallel approaches are playing a big role in terms of analyzing much larger data sets that are possible. And so if I were to show you the clustering now, I'm clustering on the basis of the RMSD values, which we have access to from the simulations. And this basically tells you after about 7 epochs, we already see the specific clustering in terms of saying, okay, this is a low RMSD state, this intermediate RMSD state, and there are 2 states that have like high RMSD values. So given the fact that we can scale it to much larger systems, it's kind of encouraging for us to see where we go. Now let's talk a little bit about how we are doing the drug discovery part. Here is where we tend to use reinforcement learning based ideas. The motivation was essentially driven by the fact that we needed to get a physics-based model into what the AI is actually doing so that the action space can be determined by what we observe from the physical system. So we said, is it possible for us to expand the compound space that we are looking at by using RL? So let's say I have these different compounds that are present. I can actually extend the fragment. I can basically do a similarity search. I can use a reaction synthesis model. It's up to you to do whatever you want, but there's the option to basically take that information and proceed further. And then we put it inside this engine, the physics-based molecular simulation engine, that allows us to take these existing structure, build the models, run ensemble equilibrium MD simulations or these adaptive methods, which then allows us to get a set of observables, which includes the coordinates, the trajectories, potential energy surface, as well as 2D images of how these compounds look like. And then you get a bunch of rewards, which includes the free energy terms that we just talked about a little bit before, the docking scores, perhaps, if you want to use them in your optimization function, as well as the RMSD values and other metrics that we think. So we can use reinforcement learning like ideas or active learning, and even hand optimization techniques like what an expert would think, so that the machine can actually learn from these actions in a continuous fashion. So for the SARS-CoV proteome, we said, can we look at relevant mutations and try and do some of these things? But let me show you an example so it's more clearer. So this is based on some expert guided fragment growth. So this is a set of known inhibitors for this protein called JAK2 kinase. And we have a known set of decoys. That means these are molecules that do not bind, even though they look like molecules that do bind. And what we want to do is move this from the part where it is supposed to not bind to some place where it actually looks like a real inhibitor. So if I play the movie, just to give you a sense of what's happening, on the left-hand side, you see the compounds. On the right-hand side, it's actually the action space of the docking, and it tells you what the docking score is on, basically it goes from all the way from -4 all the way to -20. And here is the value for the MMPBSA calculations that we have done, MMGPSA calculation that we have done. So as the reinforcement learner takes a step forward in terms of replacing some of these chemical compounds, you would see that the docking values actually start to go further and further to the left. Just let it play so you can actually see the entire set of moves. But at the end of the story, it's important to realize that we do see the MMPBSA values going down further. And then eventually it'll do something quite weird where it'll jump even more. But the idea was basically to say it is possible for us to use these sorts of reinforcement learning ideas where we can combine that with free energy computations and come back with compounds that we think are like reasonable estimates of how the binding should happen. So this is basically the final molecule that seems to bind well. And you can do the same thing with the main protease system too, where we have taken a small molecule and then started to manipulate it a little bit by a little bit so that we can actually see where these things are. So if I were to go back and look at what my action space said, this is the first iteration. And by the end of the 25th iteration, we actually reached a place where the docking scores have actually gone down quite a bit. And not only is that happening, but we are also seeing that the MMGBSA value started hitting a low point where we can see, okay, there's some compound that bound to it and it stays stable over the course of the simulation. And you can also observe the various different scores that we have been able to extract, including the terms of, the energetics that are important for characterizing how the molecule binds. And we can also just look at the pocket and see how much RMSD it has to the crystal structure. We shouldn't be doing anything weird. And so we take care of all those different types of details. But what I want to show you is basically if you analyze this data with PCA, you are able to see that we can get some amount of clustering with respect to the different portions, where you can clearly see the presence of clusters where we get low energy binding compounds versus high energy binding compounds. So this sort of allows us to explore a lot in terms of automatically placing groups of compounds and optimize the leads in some sense. Now you can ask this question of saying, this tells me that if there are similar compounds that are expanding the scope, I can basically organize this into a very large graph for optimizing these ligands and fragments. So this is a radial graph that basically consists of all these 6 million compounds from a library called as MQ, against which we did quite a bit of docking. And the nodes are essentially either fragments, that is small units of ligands, or the ligands themselves. So as you move from the center all the way to the edge in this radial graph, it's basically going from fragments to more ligand-like molecules. And edges are basically telling you what sort of transformations exist. So if there is a path from node A to B, it basically allows you to map an exact path from a fragment all the way to the node. And the nodes actually give rise to like tighter binders. So nodes that are at the edge of this are the ones that have like tight binding. But if you were to move inward, now you can actually see there are some places where you can see graphs that have like gray dots on them. And these gray dots essentially represent places where the binding is not great. But all the places where you see red dots, the energies are quite sufficient that they would bind. So you can basically kind of get rid of a lot of the space by not looking at them, because you don't have to look at these fragments at all. They will never lead you to a good binder. So we applied theory like, we applied algorithms like PageRank and started to say, can we dynamically prune this chemical space very quickly for us to identify? So this was the hypothesis which we went. And in fact, we do find that this is the 3CL Pro system that I was talking to you about. There is a binding site complementarity that you can observe from this. This is a top ranked compound that we got from PageRank idea. And you can see that the docking score is about minus 10.56 with the ChemGauss score. And this tells you that this molecule is actually forming nice interactions with the glutamine 192. There are some polar hydrogen bonds it forms with some of the other sites. And it covers almost all the 4 sites where it has to be bound. So this is something that you can find almost automatically without having to, indulge a lot in terms of searching through these spaces and saying, how do I navigate them? We can do the same thing with the JAK2 kinase. And so we started looking at these different fragments and started putting them together in some discrete format. And this is basically navigating the graph from the innermost portion all the way to the outermost portion. And the last thing that you realize is that it results in a very good binder, which is baricitinib, which is a good molecule that seemed to bind to the protein with high affinity. And what I want to point out is that there is a residue fingerprint here, which basically tells you which are the groups of residues that form stable interactions with each of these fragments. And when you basically combine these fragments together, you essentially end up getting molecules that start to unify the set of interactions. So in this final case, there is only one interaction that's left out, which is this glycine 856A, which is grayed out, but the rest of them actually form like good interactions with the molecule, and that results in good design that can be basically a very simple graph traversal based inference on these graphs. The simple comes from the fact that you're navigating on the edges, but the complexity is actually there because we have to use a deep learning model to kind of build the background of all of this information, right? This is another example where we are using the NSP 1016, very challenging binding site because there are multiple pockets that you can see. There is at least 1, 2, 3, 4, and we are trying to use multiple pockets to kind of cover the diversity of places where this molecule can go and bind. And given the fact that this is the interface of 2 different proteins, it also becomes important that we are able to target such interfaces. I'm not going to talk a whole lot about how we are building antibody designs and so on. There's a bit of work that's going on. Again, we are using the same strategy of the reinforcement learning driven optimization, but here we are starting with the sequence of the protein using Rosetta to basically optimize some of these structures. Once we get those structures, we are able to seed them back into our machine learning models. And these physics-based models that come out after machine learning actually tell us whether the interaction is going to be stable or not. And this is joint work with Ozan and Carla from Argonne, which we are pursuing right now in terms of looking at various ways in which we can refine known antibody sequence structure designs for this protein. So here is some proposed design that we are looking at. This includes all of the glycosylation that we got from Rami's team. And this kind of helps us look at different places in which we need to focus and target our attention on. So in some sense, what we have kind of outlined, hopefully, is, a lot of these discoveries are being accelerated through the use of AI in meaningful ways. Currently, what we have done is we have gotten about 30 compounds that have been initially tested. They have been successful. They have been sent out to our collaborators at University of Chicago, where some of them are doing analysis. Larger compound library of about 1,500 or so compounds on which we are running some experiments on. Both DeepDriveMD as well as reinforcement learning ideas provide new ways to integrate these physics-based models into AI, so we can think of ways in which we can accelerate the time to solution, really speaking. And one of the things that we are discovering a lot more is the fact that computational needs are getting much larger. especially because there is a deep learning algorithm or a reinforcement learning algorithm, which are harder to train or harder to get done in the amount of computing resources we might have. So there is a need to actually have integration across heterogeneous architectures that allow us to exploit both HPC and AI at the same time. And that means that there is a need to develop better workflow management systems and also come up with ways in which we can manage such large jobs. Of course, to end this whole thing, where we are taking all of this stuff is basically towards, this multi-scale modeling. So this is the coarse-grained simulations that have been driven by Carlos Simmerling group at Stony Brook, where we can see how the spike protein attaches itself to the ACE2 protein. So we have removed the virus from here. You're not seeing it right now. But this is one spike protein that interacts with the ACE2 receptor. We are trying to analyze the events that are going on as it binds to it. And this is using a coarse-grained model based on all atom models that have been built from the beginning. So the next step is to basically say, how do we map these parameters of all atom simulations into coarse grained so that we can keep them consistent using a machine learning principle? Of course, all of this is not possible without all of the people that are involved in this research, nearly about 300 people. The computing support was provided to us by Argonne Leadership Computing Facility as well as Oak Ridge Leadership Computing Facility. We were also fortunate to get compute time on TAC, which is the Texas Advanced Supercomputing Center, San Diego Supercomputing Center, as well as Indiana University. And we also thank the HPC Consortium for all the time that they've been very kindly lending us. Funding acknowledgement, of course, is from the National Virtual Biotechnology Lab. There's internal funding from Argonne, as well as the exascale computing project for cancer that we have been working on currently. So with that, I'd like to thank you all for your attention. Please feel free to ask any questions that you might have. You can also direct questions to me at my email ID that's shown here, or you can chat into the questions tab, and I'm happy to answer any questions. So thank you very much. I look forward to your feedback.
Thank you, Arvind. That was a very interesting talk. Lots going on. A lot of it probably went way over my head, but very impressive. So I'll just wait for some questions to roll in. So if anyone has questions, please type them into the questions box and I'll read them out. I had a question about the coarse-grained modeling of the spike protein that you mentioned at the end. So sure. At the moment, what force field is that being done with? Is that Martini?
No, unfortunately, it's not Martini.
Okay.
Or I don't know how to say this more politely. I mean, the way we have been using this is there are learned force field potentials that we are using comes from Amber simulations. Okay. And then we are trying to come up with beat definitions on the fly.
I see. Okay.
And this is some work that was pioneered by Cecilia Clementi's group and things like that. People have done quite a bit of work in going there. But what is different in our approach is that it's actually coming from the sampling that's being done on a single spike where we know which are the different conformational states. And we use that information to kind of integrate how the different motions will happen and how we can capture that into our ML model and then build the beat definitions based on it.
I see. So would the conformations that the protein can take on in the coarse grain model be in some way restricted then?
It's probably more than restricted. I would say it's informed, right? It's not constrained. Okay. We don't constrain, but it is informed by the different states that it can take a look at.
I see. Okay, so you're just jumping from most probable state to most probable state.
Exactly.
Okay.
And ultimately, the coarse grain model, when we are running the dynamic simulation, it might propagate elsewhere, which is another problem in itself, which is to maintain the consistency between all atom and coarse grained data, because we won't be able to track those two situations automatically.
I see. And how how coarse is this coarse graining? What's the rough atom to bead mapping that is being used?
It depends on where you are on the secondary structure.
I see. Okay. So
Unfortunately,
it is truly multi-scale in that sense that you'll have some parts that use a
yes.
larger atom to bead mapping. Okay.
And
Very
as we're running these things, we are also finding a lot of trouble, just to be cautious about the whole situation, right? Once I say this one, then there's this jump of saying, how do you manage the time and what sort of, things that you take care of in terms of jumps and things like that.
yeah.
We are finding that it's a little choppy right now, right?
I
This
see.
is not that smooth right now. I wish it was, but the hope is that these continuous learning representations that we are building would be useful or would be directed towards that sort of a thing where we can smooth out these transitions in a much better way. It's
I
not
see.
there yet. We've not done
Okay.
that work at all. We are still thinking and working through it, but this is something that we would definitely want to do.
hmm Sorry to ask you questions about that tiny little bit at the end.
Absolutely.
So I'm just going to check if I'm not seeing any more questions or any questions coming up in the box just yet. Oh, we've got one. Okay. So this is from Christian Margreiter. I hope I'm saying that correctly. Great presentation, very impressive work. You mentioned you integrated other docking backends than OpenEye as well. Did you benchmark them?
Yes, we have done some amount of benchmarking in terms of their reciprocity, so to say, in terms of how good the binding poses are. The energetics are a different story, though. I'm sure you know that in the sense that the docking scores and the energies that we get are not exactly the same, and therefore there are some issues with it. But the docking poses themselves do match up very nicely, at least in the cases that we have looked at. There are some cases where they don't, but it's kind of encouraging that we are able to use multiple docking engines in some sense to kind of inform where we should be going. Dominantly, we have been using OpenEye because we just have a preference for it, and it's also like really scalable in terms of how we can run it across all these protein sets and across different systems. And so we prefer using that. But we have also done it with Autodoc right now, and there's work with Doc that's also continuing where we are getting some more results from them. And we hope to be able to release all of that data outside. So everything will be available online.
Okay, great. Thank you. I had another quick question. It was about the, so it was on the slide with the animation on it where you showed the molecule starting off as a decoy. So I just wanted to check that I understood this correctly. So you start off with this decoy molecule, and then as you're going along to improve the MMGBSA minimization score. So is what the algorithm doing, is it actually growing the molecule? Is it switching out functional groups?
Yes. So if you pay attention closely, so at the bottom, you can actually see what groups are being replaced.
Oh, yeah. Okay.
This
very, very cool.
There you go. This was the starting molecule. This is the second molecule it tests. Third, fourth.
And are there any sort of checks that are there to make sure that these molecules are synthesizable? Or is that something you have to assess at the end?
The synthesizability is an interesting question. We are looking at that. There's a group that we are currently working on in order to include the synthesizability aspects of it. But when I showed you the graph, right? When I showed you the graph, these are all neighbors that are coming out of this functional groups that we started with. So each molecule, if you take a big molecule like baricitinib, right, you can actually break them down into their subconstituent fragments. 1, 2, 3, 4, and however you want to combine them. And if you were to walk on this graph based on those fragments, you ultimately end up with baricitinib here. But there is a path that actually takes you from one of those fragments all the way to baricitinib.
I see.
And there might be other groups that, there might be other groups that equally interact well and so on. We can discard them. But we just gave you an example where we wanted to reconstruct what happens with respect to baricitinib.
I see. Okay. So that will be something you will be looking at implementing at a later stage to make sure that you have viable synthetic steps.
Correct.
Combining them. Okay.
Good.
Very interesting. We've got a follow-up question from Christian. So follow-up, except for the data, are you also releasing the code base?
Yes, absolutely. We are releasing the code base. We are releasing everything that we have done in terms of the setup and how this is run on our supercomputers. I mean, of course, there are some limitations and caveats because we expect the data in a certain format, but we have tried to keep it in a way that is consistent with how we did the data release for our machine learning approaches, right? So the larger data set release that we had just now, which is about 60 terabytes of data, all of this is represented in some canonical form, and this is how the data would be available, which is some canonical form which we are still working on.
Okay, thank you. Well, we haven't had any more questions in, so
perfect.
I think it's probably a good time to wrap it up now. So just thank you again, Arvind, for your presentation. It was very interesting. And thank everyone. Thank you to everyone for attending. And we will be uploading the talk on YouTube and the COP Biomed website soon, so you can go back to it and pass it on to friends and colleagues. All right. Thank you very much, everyone.
Thank you. Thanks a lot.
Bye.
See you then. Bye-bye.